my name is Jessica Fairley. I'm obtaining a Bachelor of Science degree in Molecular and Integrated Biology. I started working in Dr. Catherine Brissett's lab towards the end of last semester. Since then, I've been assisting postdoc Timothy Caselli and reviewing literature. I've not completed enough primary research of my own for this presentation, so I decided to present a literature review. This Lyme disease review will cover pathogenesis of disease, clinical aspects, and epidemiology in the clinical outlier debate of post-treatment Lyme disease syndrome. First, we'll look at the overview of Lyme disease pathogenesis. The ability for Lyme disease to be a recurrent zoonotic disease despite only having indirect means of transmission is initially shocking, and it is the #1 vector-borne disease in the United States. For a quick overview, Lyme disease is a bacterial infection of Borrelia burgdorferi sensu stricto. It is a zoonotic disease that is indirectly transferred from infected host to tick, then tick to new hosts like humans. The Ixodes tick, also known as deer ticks, is the vector that propagates transmission from host to host. The wide range of Ixodes ticks and number of Borrelia species allows the disease to have a global presence, which is why it is found on every continent except Antarctica. Some interesting statistics. So there are approximately 300,000 cases of Lyme disease reported nationally every year. The right picture from the CDC shows a representation of this number by placing a dot for every reported case. In addition, according to the CDC, it is the 6th most common of all nationally notifiable diseases. At the bottom is the reported case of Lyme disease by state. We can see that North Dakota has considerably lower Lyme disease cases than compared to Minnesota, but that is largely due to our smaller population size. So Lyme disease has a variety of signs and symptoms. When it becomes more of an immune response, you'll see more flu-like illnesses. However, reports show that only approximately 50 of people who contract Lyme disease don't actually realize they were even bitten by a tick, and they just find out through diagnostics later. And only 42% of Lyme cardiodactylitis cases develop a rash, like you see with the classic Ehrlichia migranis. Other symptoms include flu-like symptoms, nervous system abnormalities, joint, tendon, and muscle pain, which is mostly due to the immune response. So what causes Lyme disease? Well, it turns out to be a bacterial infection of the spirochete bacteria under the genus Borrelia. Currently, Lyme disease agents are often grouped together and referred to as Borrelia burgdorferi sensu stricto. However, this is just a categorical group name for 18 different species found globally. 3 most pronounced species with the highest known infection rates are Borrelia burgdorferi, Borrelia abszelii, and Borrelia gruenii. Borrelia burgdorferi is the most prevalent species in the United States, while Borrelia abszelii and Borrelia gruenii are the most prevalent species in European countries. Picture C shows a scanning electron microscope of Borrelia abszelii. The picture D shows a little passage, Borrelia burinius, which is also a North American strain of Borrelia, and it was isolated from the cerebral spinal fluid and cultured, and is labeled with fluorescent anti-outer surface proteins. And in my experience, from looking at Borrelia under the microscope, they are highly representative. So we're going to look at some clinical aspects as well, and we'll just dive on into it. So the diagnostic and testing There's a variety of ways to do it. If you want to do something quick in a laboratory type setting, you're probably going to do something like an ELISA. However, this has a 20 to 30% false negative antibody positive test, which is due to the process in general, because for an ELISA, they use general isolates of the immunoglobulin proteins. So they'll be using different proteins in the beginning of an infection versus later infection, which can cause some issues with that. Otherwise, you can do something more specific, like a Western blot, where you target certain proteins. PCR, it's a similar thing with the targeting proteins, and you can also culture Borrelia from the blood. And then towards the right, they show some drugs that treat Lyme disease. For adults, you'll commonly be treated with amoxicillin, and for kids under 18 years of age, doxycycline is prescribed to them. And then also, if one of those two don't work, they also use cefalomycin, but that's mainly for adults as well. So the immunological response starts with the Ixodes tick's hypostome anchoring into the skin of the mammal, bird, or reptile. It is then it releases saliva into the feeding pit to degrade surrounding tissues and to create a blood pool meal it takes in. In this process, bioactive agents that ward off infections and Borrelia are released into the feeding pit. These bioactive agents are intended to block the innate immune response, is chemokines and cytokines that elicit an immunological response. SALP15 and Celstine L are a couple of the bioactive agents important for helping Borrelia to establish an overwhelming infection for the host. Celstine L blocks neutrophil chemotaxis response that under normal conditions would prevent bacteria from entering the bloodstream, while the SALP15 inhibits the CD4 plus T cell activation. And interestingly enough, Borrelia are able to cloak themselves from the CD4 plus T cells by binding to SALP15 to their opsin C protein coded on their body. In addition, they conceal themselves from alternative complementary pathway by using surface lipoproteins to bind complement factor H proteins. By this time, the infection is overwhelming for the host, so maintenance hosts like small animals, birds, and reptiles who have co-evolved with the Borrelia are able to live the rest of their lives with an infection and still remain normal functions. However, for humans and other domestic animals that have not co-evolved with Borrelia as long, and they tend to be dead-end hosts, the Borrelia will eventually get recognized by the immune system, and an immunological response is then elicted. By this time, the bacteria have successfully established an infection, and the immunological response is not enough to clear the infection, which is why humans and domestic animals will need antibiotics to clear it. So the epidemiology. So this is a main factor for host pathogen relationships, especially when we're talking about a vector-borne disease. So there are several Ixodes ticks worldwide that can carry Borrelia, and these ticks, especially the nymph size, they tend to be the size of a poppy seed, so they're really small. And they have a 2-year lifespan. So in the spring of the first year, the eggs will appear, and then by the same summer, the eggs hatch into spirochete-free larvae, and there it is. They'll feed on their first host, and this is where they can pick up the Borrelia bacteria, because they're not naturally born with it. It's not a vertical transmission. So they have to get it from their maintenance host, which is small mammals, birds, and some reptiles, but not many. And then the fall of that first year, they drop to the ground and molt into nymphs. And then by the spring of the next year, because they tend to be dormant during the winter, they will feed on a secondary host. And if they have the Borrelia, they will infect that host. And if they don't have the Borrelia, they can still contract it from their host. And then during that second year, they can travel to larger hosts, and they'll tend to feed on larger mammals or people and domesticated animals, which would be their dead-end host. But for like deer and stuff, they can remain a high enough infection of the Borrelia to actually infect the adult ticks. And then that same year in the spring, they will lay the eggs, and then they will pass on. And then the cycle starts over and over. So the host and transmission, we do see, we can map out a phylogenetic tree based on the species and vectors. So we'll notice that we see our Borrelia bigdorifri, senso stricto there. Although that encompasses all of them, I think they just meant to put Borrelia bigdorifri, the American strain. And all of these up here are the American Borrelia strains. And we'll notice that their main Ixodes ticks that they occupy are Ixodes ricinus, Ixodes scapularis, and Ixodes pacificus. And then as we move down, we tend to get into the European strains, which are right here. And their main vector species for ticks is Ixodes ricinus and Ixodes persisculus. And then down here we get into the more of the Asian strains. You'll find these two more closely related to closer to Japan, and these are more in deep Asia. And it's interesting because they didn't look at all of the continents, which is funny, and I kind of went into more research about it. But I guess Australia, they've been able to show that Lyme disease is present in individuals, but they have not cultured any of the bacteria from their patients, so nothing has been identified, and their vector species is also identified. And it doesn't include all of the species, because like I said, there's 18 different Borrelia species. And for example, Borrelia miyamotoi, which was first identified in Japan in 1995, was also detected in North American ticks, especially in the Ixodes pacificus and the scapularis, which is interesting that it was able to transfer or travel from Japan to the States. But I think we just live in a globally connected world, and it's a lot easier for that to happen. So geography-wise, the Borrelia bacteria tend to be concentrated in the northern eastern hemisphere of the United States. And that could be due to a variety of reasons. It could be environmental factors, it could be population factors. If you have crowding, it can create pretty much a breeding ground for bacteria of such. It's easier to get to one host to another. And then in general, the world view of it is really interesting. And you'll notice that there are some white spots here, and that's mainly due to the fact that there isn't a whole lot of research of Lyme disease in those areas, as well as there isn't a whole lot of healthcare access in some situations where they're not actually getting tested for Lyme disease. Because like I said, Lyme disease can present a bunch of different symptoms, and a lot of them don't necessarily point to Lyme disease right away. It can be identified as just like depression or arthritis and such. So some environmental factors for the Ixodes ticks in general, just because they're the main vector, because they are the vector for Borrelia. It's really important to look at how their environment influences them. So laboratory conditions do not accurately reflect the conditions faced by exotic sticks in nature. So they are looked at environmentally realistic values. In figure 4, up at the top here, they experimented the effects of temperature and light dark cycles in a moderate relative humidity, which is at 85%. They also took the mean temperatures in light and dark cycles from the northern and southern conditions and analyzed exotic ticks under north and south conditions. You'll see that the blue is the